Good afternoon, everyone. I'm David Hahn, the Craig-Burge Dean of the College of Engineering, and I would like to welcome everyone to the third and final installment of our Fall 2021 Faculty Lecture Series. Our goal remains the same, to share research success and impact with our alumni and friends, many who generously support the college and have truly helped to make this happen. As it's homecoming week as well, I want to give a special welcome and greeting to our college alumni. Our football team may be struggling a bit, but I'm happy to report that the college is thriving. Go Cats. Today's talk is entitled Medical Sensors. I'd like to set the stage for today's discussion with some numbers that I have shared before. In 2019, the US healthcare spending was 17.7% of our GDP, reaching $3.8 trillion or $11,582 per person. As a reference, the average across the European Union is right at 10% of GDP for healthcare. It's clear that we do spend a lot of money on our health. Today, we're going to explore the use of technology for novel, advanced, or point-of-care biosensing that does promise to revolutionize healthcare with low-cost and scalable solutions. Our faculty research efforts are focused on a new paradigm of photonic-based sensing, including the use of common household devices in combination with novel sample interfaces. You know, President Robbins often talks about the fourth industrial revolution, which includes the use of the Internet of Things, integrated automation, communication, combined with AI and machine learning. With novel health sensors, the monitoring of pathogens and biological molecules can provide real-time data for the diagnosis of disease or tracking the spread of viruses. Today, you will hear about two exciting faculty-led efforts to advance these technologies with a wide range of applications around healthcare sensing. I'd like to introduce our two panelists now, if they would join me. Good, welcome. There we go, thank you. Welcome. So first I'll start with Dr. Judith Su. Judith is an assistant professor in biomedical engineering and an assistant professor in optical sciences at the University of Arizona. She's also a visiting associate at Caltech on the editorial advisory board of Translational Biophotonics, a member of the scientific advisory board of femtorays technologies, and on the board of scientific counselors for the National Institute of Occupational Safety and Health, NIOSH. Judith received her BS and MS from MIT in mechanical engineering and her PhD from Caltech in biochemistry and biomolecular physics, truly interdisciplinary training. She will be giving a keynote talk at the SPIE Photonics West 2022, one of the larger biophotonics meetings in the country, and is on the technical program committee for CLIO 2022, and will serve as the program chair of OSA's 2022 Integrated Photonics Research Conference. Very engaged in their community. Her background is in sensing, imaging, microfabrication, optical instrument building for biological and medical applications. She is the recipient of an NIH R35 Outstanding Investigator Award and an American Society of Laser Surgery and Medicine Young Investigator Award. She is currently a Scialog Fellow, a senior member of the Optical Society of America, and a full member of Sigma Chi. Her work has been funded by the NIH, the NSF, DITRA, the Gordon and Beatty Moore Foundation, the Flynn Foundation, Partnership for Clean Competition, Arizona Alzheimer's Consortium, Cargill Incorporated, and the University of Arizona Cancer Center. Welcome, Judith. Our second panelist today is Dr. Jeong-Yeol Yoon. Jeong-Yeol received his PhD in biomedical engineering from UCLA and joined the University of Arizona in August 2004. He is currently professor and associate head for graduate affairs in the Department of Biomedical Engineering in our college, with additional appointments in the departments of biosystems engineering and chemistry and biochemistry. Jeong-Yeol was the president of the Institute of Biological Engineering in 2015 and currently serves as the editor-in-chief for the Journal of Biological Engineering, associate editor for biosensors and bioelectronics, and is on the editorial board for scientific reports and several other journals. He has authored 2 textbooks on biosensors, published by Springer and Elsevier, and the 3rd book on tissue engineering is in the works. Dr. Yoon's research includes work on biosensors, and he is widely recognized for his contributions to smartphone-based biosensors and medical diagnostics. Welcome, Judith. Welcome, Jeong-Yeol. We're glad you're here. Before we get into your presentation, your formal presentation, to set the stage, let's start with kind of a big picture question. Looking at our educational mission, how might institutions such as ours keep pace and prepare students for work and advanced healthcare devices sensing beyond their university days, including supporting local government or commercial ventures? And I do note that Tucson is home to many optics-based startup companies. So I'll go to you, Judith, and then to Jeong-Yeol.
Well, thanks for the introduction. So I think that's a really great question. I think, when you have a world with rapidly changing technology, I think it's important to, educate students with a solid base of fundamentals such that they can, specialize rapidly without being specialists. And I know that you had mentioned that Tucson was home to a lot of optics companies, many of which were founded by U of A alums themselves. So I think it's also important to connect with alumni such as, the people attending today, for the opportunities and knowledge that you can provide.
Excellent. Jeong-Yeol, you're muted.
Yep, sorry about that. So in my personal opinion, I think it's really important to utilize our senior design projects to connect our undergraduate students to work with the industry people out there. So that's going to be a really great opportunity for our students to get better prepared. But at the same time, faculty members also work really hard to connect with our alumni out there and industry people out there to establish collaboration and then potential investment and research projects, or maybe we can jointly ask for federal funding. And so we can actually work together as a team and then get our undergraduate and graduate students involved in those projects to get better prepared for their future career. And so that's translation and commercialization. And we need to educate them how they can the students actually have a limited understanding about how industry works, like how we can recruit the people, how we can gain investment funding, and so on. So the entrepreneurship type classes is also important for many engineering students.
Outstanding. We certainly have two outstanding ambassadors to the community in both of you. So I appreciate your answers and your thoughtfulness of willingness to engage the greater Tucson community and see that technology is transferred nicely. So what we're going to do now is have you each give a short presentation to kind of set the stage for our Q&A. So we're going to start with you, Judith, and then when you're done, you'll unshare and go to Jeong-Yeol. So Judith, take it away.
It's a great pleasure that I'm here today to share with you guys the work that I'm doing in my lab. So our lab specializes in developing ultra-sensitive sensors for a wide variety of applications, but one of our main application areas is medical diagnostics. So as I just mentioned, our lab specializes in developing ultra-sensitive sensors. And so the sensor platform that we used is shown in this picture on the right. It's called the microtoroid optical resonator. It's essentially a glass donut. It's about the width of a human hair. And I've put this red blood cell next to it so that you can get a size comparison. And essentially what we do for our medical diagnostic applications is we detect biomarkers of diseases in any sort of bodily fluids like blood, urine, sweat tears, basically anything that sort of contains a biomarker for disease. And we have a lot of applications in our lab. I'm just going to go through a couple of them. So one of our main application areas is early detection of cancer. We also work on early detection of Alzheimer's disease. We just finished a project a few years ago on performance enhancing drug detection. We have a large project with the Department of Defense on chemical threat sensing. We look at things like water safety, food safety, environmental monitoring, and then a large portion of my lab focuses on further improving our sensing platform, for example, by making portable sensors more robust sensors. So our sensors are called optical whispering gallery mode resonators. They're named after the acoustic whispering gallery phenomena, and perhaps the most famous example of this is St. Paul's Cathedral in London. So in St. Paul's Cathedral in London, underneath the dome, there's a gallery known as the whispering gallery. And if you stand at one end and you whisper in the gallery, your friend can hear you 40 meters away at the other end because sound travels along the edges of the wall of the gallery with negligible loss. And so here's on the picture on the left, here's an example I like to use because you can really see this curved wall. So this is called the Echo Wall. It's in the Temple of Heaven in Beijing. I'm in the front and my husband's in the back. And he can hear what I'm saying, even though we're far apart because sound is traveling along the edges of this wall. And so our sensors operate on the same principle, except instead of sound traveling along the edges of a wall or some architectural feature, we use light that's traveling around the edges of this sort of glass donut. So we're the optical analog of the acoustic spring gallery. And so here's an artistic rendering of what our sensor looks like. The bright colors represent the light within the device. And so you can see the light is traveling along the rim of the device, and it travels via the principle of total internal reflection. So you can see this is sort of similar to how light travels through an optical fiber, but you can think of this as sort of an optical fiber that's sort of bent back upon itself. And so the light is continuously trapped in this ring, essentially. But the light is not perfectly confined in the ring. Some of the light extends a little bit outward beyond the periphery of the ring. And this is what gives our device such high sensitivity. So what we do is, this is made of glass, and so we can functionalize this with capture agents for various different biomarkers, for example, like antibodies. And when things bind to the surface, the light continuously goes around and interacts with it. So it constantly re-interrogates our sample. And this is what generates a very high signal for us. And you can see a little GIF of this on the right. So this sort of vertical rectangle represents a waveguide, which is what we use to couple light into our sensors. And then you can see this constructive amplification of light within this device. And this happens at a particular wavelength known as the resonance wavelength. And so why would we use these sensors for biosensing? So we have a number of advantages. So there's a picture of this on the, on the right, in the upper left-hand corner of this image. And then the bottom circles are the sort of the top view. And so you can imagine that we could coat each one of these sensors with a different capture agent so that we could look at different biomarkers, and so we could eventually examine panels of biomarkers. And so our sensors have a number of advantages. One is that you have the potential for very high throughput measurements because you have on-chip fabrication. So we can make hundreds of these devices. We have a very, very small sensor, which means that a very, very small sample volume is required. So less than 10 microliters, which is about a drop. So we just completed a study on ovarian cancer, which we were able to track tumor progression using just 4 microliters of a very small vaginal lavage sample from a mouse. We can get real-time measurements of binding speed. We're very sensitive. We're down to single molecules. So we're label-free, which means that we don't have to put any sort of radioactive tags or fluorescent labels on what we're trying to sense. And we could obtain results in under 30 seconds, which is very important for some of our applications, such as infectious disease detection. It's very, very important to be able to get timely detection. And so this is one of our first applications that we did, which was on a minimally invasive tumor biopsy. And so what we did is that we implanted mice with human Burkitt's lymphoma tumor cells. So this cell line was picked because it's a very aggressive tumor. And so we were looking at this particular biomarker called exosomes. So exosomes, you can think of it as little bits of the membrane of your cell. And what's exciting about exosomes is that when you have little bits of the membrane of a tumor cell, it carries little bits of your tumor inside your bloodstream. And so the idea is that you can exploit the circulation of blood to detect if a tumor is present and how far it's progressed without having to do surgical intervention, without having the need to find or access a tumor and do a biopsy. And so this is really good because people are very often, they're interested in, they're willing to, for example, be biopsied once so that they can find out what they have, but they're more reluctant to be biopsied multiple times. And so this is a good way, if you can get something like this to work, where you can track tumor progression. You can also look at treatment outcomes, for example. So how do different treatments affect different biomarker levels? And so the way to interpret this graph is you can see, so the y-axis or the vertical axis here is the signal from our sensor. So you can see from week 0, we don't see much signal from our sensor, but then as each week progresses, you can see larger and larger signal from our sensor as more of these exosome biomarkers are binding to the surface of our device. And then the control experiment for that, of course, is a mouse with no tumor. So we took a mouse with no tumor, and then we looked at the signal from our sensor from week 1 and week 5, and we didn't see this similar increase in signal. And so where's the future of this going? So our goal is early identification of disease prior to physical presentation of symptoms. So we want to avoid a situation where you go to the doctor because you're starting to feel like in extreme pain, and then you're being told that you have stage 4 cancer and you only have 6 months to live, something like that. Because we have a very, very sensitive sensor, we can detect very, very low quantities of these biomarkers. So we're interested in making these devices portable point-of-care devices, either handheld, wearable, or both. We're interested in making this as prevalent as a blood pressure machine in a pharmacy or mall. Our time to getting this into the hands of the public is hopefully in less than 10 years. Our technology is already fully licensed by a company called Femtores Technologies, which is a startup company based in Italy. And I would like to thank our lab and all of our sources of funding. And I'm happy to answer any questions that people may have.
Thank you very much, Judith. Before we jump to Jeong-Yeol, let me just ask one point of clarification that someone just posted. On your mouse model experiment, you're not putting your sensing device in the mouse, you're drawing a blood sample and bringing it to your device. Is that correct?
Yes, that's absolutely correct. So we are not an in vitro sensor. So we're outside, sorry, we're not an in vivo sensor, we're outside in the laboratory. And then we just flow any sort of biomarker containing solution over our devices.
Got it. Thank you for that quick clarification. Excellent. If you unshare your screen, we'll turn it over to Jeong-Yeol to share his screen and give us a presentation on some of his exciting biosensing work. Over to you, Jeong-Yeol.
Thank you. Can you see my screen okay?
Perfect.
Sounds good. So hello, my name is Jeong-Yeol Yoon from Department of Biomedical Engineering. So I have been known as a smart fun biosensor guy in scientific community. So that's the topic I'm going to talk about today. And obviously, this can be used for the medical healthcare applications, but equally can be used for environmental and public health applications as well. So my background was microfluidics and biosensor when I did my PhD study at UCLA. And I came to U of A in 2004 and continued working on this. And then it's primarily optics-based microfluidic sensor that can be used in vitro, I mean the outside of your body. So basically testing your blood, testing your urine, testing your nasal swab, testing your saliva, and sometimes testing your feces, and so on. But it's primarily either urine, blood, or saliva. And then in late 2000, the smartphone came out, specifically iPhone. And then I found that the sensors, there are many sensors already incorporated in the smartphone. So here's the next slide. We have the front and rear camera, IR camera, proximity sensor, and GPS, accelerometer, gyroscope, the light source, white LED flash, and then it has a computing power, and they send your data through Bluetooth, NFC, Wi-Fi, 4G, 5G network. So can you utilize this? So the first project that I worked in collaboration with industry was create an optical reader device based on your smartphone. So this is the lateral flow assay, also known as the rapid kits. And if you do not know what they are, think about pregnancy test, or more importantly, the COVID-19 rapid antigen test. It's all based on antibody. So we're just gonna take up your blood or saliva sample, add it, and then the band will develop. And so you can check the positive or negative with your naked eye, but you can actually insert it into the optical reader device to quantify the concentration. And then that device tends to be rather expensive. So what I did, I replaced that optical reader device, typically cost around 20,000 to 50,000 using your smartphone. So in that case, I need the optical reader device printed in with the 3D printer. And then we did a lot of optimization. So this is primarily angled detection. We are measuring at a certain distance and angle to maximize the assay. And then we coded our own app. And then we demonstrated 1 order of magnitude better limit of detection in comparison to the standalone lab, the laboratory optical reader device. So we got a lot of media attention in that year. And then we actually moved on to a different application. So if you can do that on the commercial rapid kits, can you do that on paper microfluidic chips? Whereas the microfluidic channels are being printed on your paper, and we can do multiple assays in a more complex manner. But this time, we are measuring our signal at an optimized angle and distance using the smartphone's gyroscope and the accelerometer. So it worked quite well, and we called it as a smart paper app. And we got a publication in 2013 as well. So these are all based on antibody assay. And if you got the positive signal with this antibody assay, then you may want to test it for this genetic sequence known as the PCR, or polymerase chain reaction. And of course, PCR requires the loss of apparatus, trained personnel, and then it usually takes a full day. And we actually created the PCR device that can be run in a small form factor and then coupled with the smartphone optical reader. And then we made a lot of innovations on this device so we can finish our assay. From sample to answer, it took about 20 minutes. And then thermal cycling itself takes about 3 minutes compared to several hours in the laboratory. So I'm gonna share this screen. So this is how our device works. And you can clearly see that all those detections are being made by the smartphone. So we made an equal the contribution to the PCR assay as well. So once we have demonstrated that, then we wanted to push our limit. Maybe can I detect the target molecule, in this case, norovirus, down to a single copy level? So this is almost like a molecular level detection. And to make it happen, I needed fluorescence microscope. So benchtop, really expensive microscope, so we can count those individual virus particles one by one. And then can you do that with the smartphone? And what it did is using the off-the-shelf components and assemble them all together. And then we can create the smartphone fluorescence microscope that can count your norovirus sample from environmental water samples. I'm gonna play this movie. And this housing contains the white LED light source and many optical filters embedded, so we can convert them into the fluorescence microscope. We are doing two pipetting, one with the antibody conjugated particles, the other with the water sample. And then the all those image analysis is actually done over cloud. So so you actually see analysis is being done on the smartphone, but the calculation and analysis are all done on the remote server. So we got a lot of media attention on this one as well. And obviously, when we published this paper, we got COVID-19 pandemic. So we wanted to adapt this technology for COVID-19 assay. So the device got even smaller and then simpler. And you are using saline goggle samples collected from our students. The method established by my collaborator, Michael Warby, here at the U of A. And this is our clinical data. And then all positive samples are classified as positive. And most of your negative samples are classified negative, with the exception of 2 samples. And we believe these 2 are actually positive. Maybe the students got exposed to COVID-19, but the virus load is really low. So the PCR cannot really detect them, but we can detect them using our method. And then we also, since our detection is so sensitive, we can detect directly from aerosols. So we are not collecting samples from the humans, but we are collecting samples from the aerosol passively collected on your paper microfluidic chip. And we can still do that detection. And finally, the one we are working on is that I want to develop a biosensor that requires no antibody, no enzymes, no DNA or RNA, but use machine learning to sense anything. Because for many biosensors, you need to know what you're looking at. So you can prepare antibodies or enzymes or DNA. but can you have a universal biosensor that will tell you based on your pattern, then figure out what do you have in our sample. Right now, we are using it for identifying bacterial species, but eventually we want to expand it towards the many different virus detection. I'm going to skip the last slide. And so this is the lab photo that has been taken last year. So I'd be happy to answer any questions you may have. And thanks for your attention.
Excellent. You can unshare that. Thank you both, Judith and Jeong-Yeol, for some really nice presentations. So we're going to move into the Q&A portion. I encourage those in the audience to use the Q&A chat. Judith's already answered a couple. I've pulled questions that were pre-submitted by the people registering, so I'm going to start with those and work our way through and pick up as we can in real time some additional questions. So one question is asked is, what are the most challenging factors preventing these technologies that you both are working on from transferring, this technology to actual clinical uses? And we'll start with you, Judith, and then we'll go to Jeong-Yeol.
So for our specific case, it's this use of this optical fiber that we use to couple light into our devices. So this optical fiber is really, really fragile. It's, around 500 nanometers in diameter. And so it's easily broken, and it has to be precisely positioned next to our sensor using a nano positioning stage. And so what we've been doing is we've been working on ways to do alternate light coupling into our device, so to eliminate the need for this optical fiber. And so once we can eliminate that, I think that's our main impediment to translation out of the laboratory.
Okay, very good. Jeong-Yeol.
So my immediate most important concern would be, we are using the commercial smartphones, and they have their own, the IP rights. And so we develop a lot of different things using the smartphone, and then we're not really asking their help. We develop on our own, but sometime later they just step in. Oh, this is ours. You cannot practice this. And then that's the biggest part I'm really concerned about. So I think some people are I think the Android phones are a little bit more open architecture. So so in the past, I'm a big fan of the iPhone. So I have been using iPhone all the time, but maybe I need to switch to a more open source smartphone architecture. So that's the one direction we may want to pursue. But the other direction would be forget about the smartphone, create the Raspberry Pi based camera and the Raspberry Pi detection device. Then we don't really have to worry anything about the commercial things. So I think that is the biggest bottleneck. And the second bottleneck is that we may need to get an approval on the smartphone itself, because if you're using a smartphone, your medical diagnostics, it may be considered as a medical device. Then they must go through all those the FDA clinical trials and approvals. So
excellent. Yeah. Thank you, Jeong-Yeol. I'm going to stay with you on a somewhat related topic, which is, we know when we again talk about medical devices that HIPAA imposes strict regulations with patient healthcare data. So again, as you move forward, transitioning your really novel technologies, do you have additional HIPAA compliant concerns, cybersecurity, those types of things when you're talking about using a patient's cell phone to record, patient health data?
So Judith, you want to go first? Just me for me?
I'll stick with you on that one since you're the cell phone person.
Okay, sounds good. So in my personal point of view, I do not want to deal with the insurance companies. As long as I can get an FDA approval, then I'm gonna probably put I really want this price tag, something like less than the copay you make. So if your copay is $30, I'm gonna make all of my devices cost less than $30. Then you don't have to worry about insurance. And the problem is we're gonna face some resistance from the existing medical industry, or maybe there's some regulatory guidance. But you know what? Back in the old days, when the pulse oximeter first came out on the market, when the first glucose meter came out on the market, the regulatory, and many people actually raised the concern about how can a person purchase this device and they read this data and interpret in his or her own, and that's basically infringing your medical practice. So this is unacceptable, and we need to prevent them from happening. Well, what happened right now, you can purchase the pulse oximeter and glucose meter without doctor's prescription, and they're really cheap, and then they can just read and interpret all the data. So eventually that's going to happen, but I really want to make it happen much earlier than, maybe in the next 5 or 10 years. So that we don't have to worry about the insurance regulations and the doctor's prescription and doctor's order and so on.
Excellent.
Mhm.
Thank you for that. Judith, I'm going to go to you now with an interesting question. You're very much versed in photonics and biophotonics. So this is kind of a future-looking question. Someone asks, will we be able to, at some point in the future, look into your mirror and have it tell us something about our condition? Sort of a, mirror on the wall, who's the healthiest of all kind of question. So I think it's an intriguing question. And where do you see that going, Judith?
I think that's a really interesting question. In fact, just the other day, I was reading a paper that had just come out where it said that, people could zoom into your eye, like a picture of your eye, and detect whether or not you had COVID with like 97% accuracy. So I think these sort of technologies, it was incorporated a lot of artificial intelligence, of course. So I think these sort of things are definitely on the horizon. And I think people are working on these technologies right now. So it might seem very futuristic, but as with the COVID example, people have started to do these things.
That's fascinating. Good. I agree with you. The idea that I know there's a lot to be done with the eyes around even diabetes and things like that of getting diagnosis. So I agree. It's a very intriguing question, and probably we'll get there at some point in time. So someone asks to discuss the issues of accuracy and drift in your devices. Is this a major issue for, as Jeong-Yeol just described, sort of over-the-counter technologies versus, devices that are prescribed by a physician and possibly used in a clinical setting? So as we bring this out to the masses, does that present problems with calibration, accuracy, and drift? So I'll start with you, Jeong-Yeol, then I'll go to Judith for her own interpretation.
Well, the calibration is a major thing because, frankly speaking, before you do the assay all the time, you need to manually calibrate them. So, for example, how I'm going to position your phone, and how I'm going to, if it is based on the light intensity detection, and if it is based on the microscope imaging, then how you need to manually focus them. So eventually, we need to create a system that are self-calibrate automatically. So that's a lot of work, and then a lot of the industrial, the developments are supposed to be they're supposed to be done on this, but it's less academic, but more commercial development at that point. But certainly, yes, it is needed, because if you're asking each individual to calibrate your machine all the time, then people are not going to use it.
Good, very interesting. And Judith, when you look at some of your technologies, how do you view these kinds of issues?
Yes, I think accuracy and drift is really important. And, we do a lot of studies to try to characterize it and minimize it. And I think there's always going to be, a difference in the quality of results you get from, a large instrument that you find in a clinic versus something that's portable and wearable at this point. So, you might have, for example, if you take a pregnancy test at home, you can get these very cheap over-the-counter tests, but then people always want confirmation from a physician where they have, more accurate tests. That's considered like the gold standard. And then I've seen that they've started to sell these sort of portable handheld ultrasound devices. But of course, the accuracy of that compared to, a big instrument that you see in the clinic. So, I mean, when you have smaller point of care devices, they might be cheaper, they might, be handheld, but I think you're going to sacrifice things like accuracy, when you start doing things like decreasing cost and size.
Yep, I totally agree. So the balance of widespread low cost preliminary answers maybe, and then maybe some confirmation on more serious issues. So that's good. Jeong-Yeol, I'm going to go back to you and pick back up on the topic of COVID. We're still in the midst of this pandemic. I know you talked a little bit about COVID in your presentation, both from the saliva tests and the potential to even do aerosolized detection. So you want to elaborate a little bit more on what are the potentials for your technology moving forward? And do you have plans to continue along those lines?
So frankly speaking, the we have spent a lot of time on developing COVID-19 assay, but our emphasis has been really, really extreme low-level detection. And right now we got the commercial rapid test. They are not very accurate, but we can detect the the presence of your COVID-19 from your saliva, nasal and saliva sample. So I think my technology has a potential use for aerosol detection, not from the human samples. That's the one area that I'm very interested. Another area that I'm very interested in is that the antibody quantification, especially neutralizing antibody. People are talking about how long you can have your protection. Of course, once you get vaccinated, you have a lifelong protection through your memory B cells and memory T cells. But if your neutralizing antibody wanes over time, then you're going to get a short infection. You're not going to die, of course, once you got vaccinated, but you're going to develop some symptoms. And people may want booster shots. And then when exactly that should be? Or do I really have neutralizing antibody? So right now, the only possible method, you need to withdraw your blood and then they get it tested in the lab. Because the antibody amount, neutralizing antibody amount from your saliva is extremely low. But I believe my technology can detect them because mine is so sensitive. So right now we have just started a project detecting the antibody from saliva. And then we don't have conclusive data yet, but we believe it is promising.
Outstanding. Good. And that's great. That's good to know. You know, that's an example looking at your data that you presented in your talk where drift is an issue, right? You have this fine line that segregated the positives from the negatives. And if that line's moving up or down, suddenly that detection is starting to come into play, right? So calibration is very important. Now, cancer remains a devastating disease, and we've had a couple of questions pre-submitted and submitted in real time around cancer. So I'm going to start with you, Judith, and then I'll come to you, Jeong. You'll see if you're doing anything in this area. But when we talk about cancer detection, Judith, what sensors are being developed, maybe using your technologies to, detect cancer? And you kind of made it address this partially in the chat, but I'll ask you to elaborate. You know, when you find a circulating tumor cell, for example, in your ex situ device, how do you then, what do you do with that knowledge, right? And how do you source that back maybe to within the body? So talk to us a little bit about cancer detection, Judith.
I think probably most of you guys are familiar with standard techniques like a mammogram, ultrasound, MRI. And now, of course, this is, a huge field. Like anyone who's doing these sort of sensors, probably they'll try to target cancer at some point. So you have all sort of different detection modalities like electrical sensors, optical sensors, everyone, trying to detect cancer biomarkers. And so personally, what we've been doing are things like what I mentioned in my talk about the liquid biopsy. So this is, I guess, what people would call a hot topic now. So there are, large companies that are doing this where you're looking for biomarkers that are shed by tumor cells, whether it's, DNA or protein biomarkers. And, people have been able to distinguish, between, so there are different biomarkers for different cancer types. So you have to find a specific biomarker for that particular cancer type. And then for certain kinds of cancer, people have been able to get, reasonable discrimination. And then it's, of course, there's all these different things like, ethnicity that comes into play for some of these biomarkers. So in our case, what we do when we detect these cancer biomarkers, like, for example, these exosomes, is we do things like we track tumor progression. So can we see how far a tumor has progressed? And then we also might be interested, for example, in evaluating things like treatment outcomes. Like, if you applied a particular kind of treatment, how do these biomarker levels change? So we haven't yet gotten that far with our cancer work, but for example, for our Alzheimer's work, some things that people are interested in are, exercise. So if you have Alzheimer's, people tell you to keep your cardio up, stuff like that. And so, things people are interested in are like, after you exercise, how do these biomarker levels change and stuff like that?
Yeah,
so
go ahead.
Oh, no, go on.
I was going to say, pull out your crystal ball and let's say 10 years from now, do you feel like, routine, you go to your physician, you have a blood draw, do you feel like, say, a decade from now, they'll be able to extract some pretty useful diagnostics around cancer early detection? Is that the right time frame to make a lot of progress?
I think so. I think companies are even doing this now, where you can take blood and you can send it out to be sequenced and stuff. Yeah.
Fascinating. That's great. Jeong-Yeol, are you working at all in the cancer space? And is that something that your technologies could have some applications to?
Yes, also I work on cancer, but I'm primarily interested in the circulating tumor cells from blood, and then also the the blood cancer. I'm very interested in detecting the cancer markers from blood, and it's all based on either the antibody-based assay, or I can also do the PCR-based gene amplification. So I work on both.
Excellent. Thank you both. I'll stick with you, Jeong-Yeol. It's increasingly common for people to use Fitbits, Apple Watches. It seems like the commercials are nonstop with the new Apple Watch about monitoring your health. Are these kind of wearable technology opportunities, can you leverage that into some of your own research interests with small sensors and those types of things?
There are a lot of other people out there utilizing the accelerometer, GPS, and gyroscope, and pulse monitoring the system towards activity trackers. Many commercial apps and commercial devices are already available. What I am interested is that they do have the image sensor. So basically, they take the image of something and then show its coloration in 3 different colors: red, green, and blue, RGB. But what if we can construct the entire spectrum? So not just like 3 colors, but 400 nanometer, 401 nanometer, 402 nanometer, keep going on, and then establish a spectrum across the entire wavelengths of your visible light, or maybe well into the UV or near infrared as well. So if you do that spectroscopy for the entire imaging, then becomes a hyperspectral imaging.
Sure.
And I really want the smartphone companies to enable that spectroscopic analysis, or maybe either hyperspectral or multispectral imaging capability. And if we can make that happen, then everything will change. We can detect the presence of bacteria from your table, from your toilet, from your kitchen. Just scan it with the multispectral imaging device, and then scan it through, and then it may tell you got some pathogens out there. And most likely it's dangerous pathogens, or it's less dangerous pathogens. So the technology is already out there. It's just a matter of incorporating them into your smartphone. And then right now the obstacle is the high price. But if you can mass produce them, it may happen.
That's great. That's some really promising new avenues, right, that could open up. So that's great. I'm going to kind of come back to both of you to our educational mission just a little bit. So you both had interesting careers and some really exciting research. Judith, you've got engineering training and biochemistry training. What are some takeaways from your professional life and career track that you would share with engineering students that are listening or will listen to this in the future that might have an interest in healthcare, and kind of maybe give them some takeaways that could guide them to contribute in this area? I'll go with you, Judith, and then to Jeong-Yeol.
For me, it's really been that the future is really unpredictable. So it's been really hard to predict what would happen in my life, 6 months from now, 1 year from now. So I would just be really, really flexible and be ready to catch opportunities when they come up. And I would also really encourage tapping into the alumni of your college. So for example, College of Optical Sciences has this huge industrial affiliates program, which has a lot of alumni, and they've been really helpful in terms of finding jobs, providing career advice, etc.
Excellent.
So I would ask I always ask my undergraduate students to participate in research activities, and regardless what their career goals are. So many people think that the research training is primarily for the students interested in graduate program or medical school. Not necessarily. I have found many of my undergraduate students successfully finding the job they want in the relevant discipline with the high-paying job. So, so the research experience is quite important for undergraduate students to get better prepared. It's very different from taking classes and then taking, they get a good grade on your other classes, but just participate in the ongoing existing project as a team player. And I also strongly recommend all undergraduate students to participate in the industrial internship over the summer. And And then there is also the research opportunity in the other institution as well. So they utilize all those opportunities, then they can be better prepared for their successful future career.
Excellent. I'm going to go back to my kind of opening statement about the dollars associated with healthcare, the 17% of our GDP.
Mhm.
Do you believe that your approaches can make a dent in the healthcare costs through some of the things that we've talked about? I'll go to you, Judith, and then to you, Jeong-Yeol.
I think definitely. I think a lot of times symptoms or what a patient tells a doctor, lacks the sensitivity or specificity to guide decision making, and this can lead to either over testing or waiting for obvious clinical deterioration to occur. And I think that our technology that we develop can do things like reduce additional testing, which is often caused by these sort of things happening. And so I think that's one way that we can help reduce healthcare costs.
Excellent. That's good. So both, low cost testing, and I like what you said about early sort of detection or waiting. You don't have to wait until things get so bad, right? And that can really have a lot of cost savings. Excellent. Jeong-Yeol, what do you feel on this front?
I would say on my own experience, because I have a lot of medical conditions, and but I receive a letter from the insurance company all the time. They do this test, do this test. And then they're doing a lot of tests, they ahead of time basically save insurance companies money, and otherwise they wouldn't do it. But the problem is this: when they ask me to do certain tests, and then there are tests that can be easily covered by the insurance, but there are other tests that is not covered by the annual checkup. Okay, you clearly see the difference.
Yeah.
And then insurance companies trying to ask all of you to get tested, but the cheaper test only. So if we can lower the price of that, the diagnostics, and then make this kits test available to everyone, that's gonna dramatically lower the cost of the healthcare expenses.
Outstanding. Judith, I asked you after your presentation to clarify that, was your device actually going inside the body, and you confirmed for the mouse trial it was not. But to both of you, do you have any future plans that you might incorporate some components of your sensors actually inside in vivo type sensing? And I'll ask you both to share any thoughts on that subject that somebody asked. So I'll go with you, Judith, and then to you, Jeong-Yeol, if you have anything to add to this topic.
Yeah, so I actually had a student that was working on this for a little bit before she got pulled away to do other projects where, yes, we are interested in sort of developing these sensors to go inside the body. And of course, that has to take a very, very different form. And we were interested in incorporating essentially little nano lasers inside the body to create sensors. And so that is an area that we are actually very interested in.
Oh, interesting. Jeong-Yeol.
And then frankly speaking, the inside of the body is not really my expertise, as you can clearly see, but I am interested in attaching those sensors to the skin of your body, so it can actually read your signal. So a good example would be the sweat sensor. So rather than analyzing your saliva or blood or your urine, but you can collect your signal from your sweat. And then that could be considered as the InBody sensor. So that's the part I'm also interested. But we also have other players here at U of A who works on the sweat sensor. It's primarily geared towards glucose sensing, but I'm more interested in the more complex molecules.
Excellent. And Jeong-Yeol, I'm going to stay with you for a second and pull a question out of that was just submitted about, your devices and your machine learning to look at wastewater, for example, following UV disinfection. So I guess the question that they ask is, can you differentiate between inactivated and active viruses to potentially assess the efficacy of disinfection or those types of applications?
Unfortunately, no at this time, because it's all based on the interaction of my certain specific chemicals or peptides with the surface antigens, surface proteins of a bacteria. So we cannot really tell whether they are alive or not. So that's the limitation. But we do have other method based on the more sophisticated antibody assay or more sophisticated PCR assay, nucleic acid-based assay. So this is more like a preliminary screening tool. So you have something, and and then we want you to further develop it. But the having the knowing what you are looking for is very important, because if you do not know what's in there, you need to test it like hundreds of different subjects. Then you don't want to do that. But this machine learning technology will tell you most likely you have this or that. I want you to further test that on these two.
Gotcha. And I'll just stick with you for a moment and pull one more question in real time. Someone asked if they've heard that some firms are already meeting with the Food and Drug Administration on using smartphones and
Yes.
apps. Have you had any exposure to those companies and working in that space, and any thoughts to add to that question?
So actually that the smartphone reader device that I have published in 2013 has been licensed out to an external company. And then they have gone through FDA clinical trials in the past couple of years. I think their product has come out on the market, the last year or something.
Wow,
So I have some experience on that. And then they're not the only one, of course.
that's outstanding.
Mhm.
Again, I'm going to take this back to education for a minute, because our graduate research program is a very important part of this college, and the recruitment of top students is paramount to the success of our graduate programs. Can you both comment on specifically sort of the attractiveness of your work to helping to recruit students? Is this a magnet? Do students really want to work on these things? So I'm going to start with you, Judith, and then I'll go to Jeong-Yeol.
Yeah, I've certainly found that many students are particularly interested in, healthcare, making an impact on people's lives. And so for them, the application of technology to healthcare is a really attractive field of study. And they always want to know, like, what classes do they need to take to be better at this, and how can they get involved and stuff like that.
Outstanding. Jeong-Yeol, what's your experience been?
Well, if you do, the impact for a walk, and then if you continue to publish, and then if you got the proper media attention, students are the ones who know the best, actually. They are quite smart, and you'll be quite surprised. And so if you do a great job, then you usually have no problem in recruiting high quality students. The downside is maybe the little bit of limitation would be recruiting students internationally, or recruiting students from the other side, other part of the nation, like for example, from Massachusetts or from Florida. They're so far away. So we need to do the concerted effort to expose our work to those people out there.
So, when I came here as dean 2 years ago, one thing that drew me was the co-location of our health sciences, our medical school, pharmacy, nursing, right here on the main campus. I'm a big believer in the intersection of engineering and medicine. Can you both speak to that in your experiences? How beneficial has it been to have health sciences and medical school right here, and maybe get into some of the details of your specific collaborations with our colleagues over in health sciences? I'll go Judith and Jeong-Yeol again.
It's been really beneficial. So I had submitted a very large proposal. It was like $6 million to the NIH recently. And one of the criteria was that, we had to be able to detect, 20 different disease types. And I was thinking, it was like, how are you going to be able to get that? So you basically, and it wasn't just like 20 different kinds of cancer. They wanted like cancer, diabetes, like a really broad. And I was able to contact people at health sciences, and we were able to get people from cardiology, psychiatry, like sort of every medical branch, and I don't think I really would have been able to do that easily at another place.
Excellent. How about you, Jeong-Yeol?
The same goes with me. For example, my COVID-19 project, I'm collaborating with the two professors in medical school and one other in the College of Science, and I am using the student sample collected by the Banner Health, and then whenever I have questions about the sample, then I can actually ask them. So how did you process your sample? And then we can deliver the sample from their laboratory to my laboratory in less than half an hour. Actually, it's about 5 or 10 minutes. And then we can just even walk our sample from their lab to my lab, which is very convenient. But if our medical school was positioned in Phoenix, we need to drive like 2 hours back and forth. And that's going to be big inconvenience for all of us. So that's definitely helpful to me.
Excellent. That's good. That's a great answer. And that's what I was hoping to hear from you both. So, we're coming to the end of the hour. It's been a great conversation. I'm going to give you, each now pull out your crystal ball and look, say 20 years into the future. What problems do you think will have solved in this area of sort of biosensing, and maybe what might the next challenges be? I'll go to you, Judith, and you, Jeong-Yeol.
So I think that, I think where this is headed is really personalized, point of care sensors. So I think you have this increasingly aging population, increasingly wanting to be more educated about their health outcomes. So I think you're going to see lots of sensors integrated everywhere. So, you might, wake up in the morning, step on your floor might have sensors to tell you about your gait and how that affects your health. You might have sensors that tell you whether you took your medication or not, and everything will be, personalized specifically to your biomarker levels, your baseline, and maybe automatically sent to like physicians. So I think it's really going to be personalized healthcare and point of care healthcare.
Wow, fascinating. Jeong-Yeol.
Yep. So the same thing goes with me. So in the future, this could be a concern for primary doctors, but I think we can use primary doctors over the Zoom, and then we can actually test our own blood or test our own saliva using the off-the-shelf the sensor devices or the strips, and the results can be sent to your doctor automatically in real time, and the doctors can see them. So right now you need to call, for an appointment with the primary doctor. You see a doctor like a couple of weeks later, then you go to the lab, and then you need to wait for another week and a follow-up meeting. Everything can be done in a matter of just one day, and then eventually those primary doctors may be replaced with the AI, artificial intelligence.
Wow.
So yeah, that's the future that people are talking about. So there will be some resistance from the the medical community and insurance companies, but since this is the way to save their time and save their money, I think it's going to go towards that direction, whether we like it or not.
I think you're going to see this democratization of science, and you're going to see more citizen scientists get involved.
Yeah, probably.
That's a great vision. I want to thank both of you for a really informative, engaging discussion. I want to thank our alumni, friends, colleagues in the audience that attended. Some great questions. This has been a really informative, and it's been a very enjoyable afternoon. So I want to thank everyone for your attendance. I hope to see many of you Friday at the homecoming breakfast for the college, or at Saturday's tailgate out on the lawn. So until then, I want to thank everybody. Wish you a great day, and see you next time. Bye. Thanks, Judith. Thanks.
Okay. Bye. Thank you very much. Bye-bye.